Okay, so today we are starting with a fascinating topic, the reproductive system. It's always fun to talk about. Why do animals have sex in the first place? To reproduce. Okay, that's one. What else? Pass on your genes. Okay. What else? Pleasure. Fair enough. Huh? Survival of the species. Wow, you all are, it's like you read the PowerPoint or something like that. Okay. So that's pretty good. So yeah, the ones we came up with in previous classes is to perpetuate the species, which is true. Okay, but remember, not all animals have sex. Can you think of some animals that don't have sex? Sponges. Some fish don't need to have sex. Most of them do. Things like sea anemones can just divide in half. Passing on genes, that's true. Okay? The whole game of evolution is being able to pass on your genes more than others in the population. But when you see two dogs going at it, do you think one of them is sitting there going, "Must pass on my genes"? No. Okay? They're doing it because they enjoy it, so that'll be on there too. To produce offspring, again, yes, that is one of the reasons that animals have sex. Do they know that they're having sex to produce offspring? Probably not. And we don't know, but probably not. To allow for greater genetic variation. We do know that animals and species that have sex tend to be more resilient to changing environments. So we think sexual reproduction is a way to hedge our bets against environments that are changing. So there's this added genetic material that can be useful in times when the environment changes. And because it feels good. Now, obviously for humans, we know this to be true. People didn't used to think about animals having sex and did it feel good for them. I took a primate class when I was in college, and we studied all kinds of primates, including bonobos and things like that. And you want to talk about an animal that gets freaky, it's a bonobo. Humans got nothing on bonobos. In fact, even on macaques, I studied macaques just for a couple weekends, and they are dirty little monkeys. They really are. So human sex is pretty tame compared to that. Dog sex, whatever, but chances are they're doing it because they enjoy it, get pleasure from it somehow. And that's important because in biology and evolution, there's what we call ultimate causes and proximate causes. Now, ultimately, the reason that animals reproduce is to pass on their genes, is to perpetuate the species, is to produce babies. But it also has a proximate part. The reason that individual animal is having sex with that other animal is because they have an evolutionary drive, or maybe just feels good, okay? Because we're wired that way for evolution to make things feel good that will help to pass on the species and help pass on the genes. Okay, so before we go on to the sexy sex stuff, we're going to talk more about the cell division that's involved in the production of gametes. Now, what are gametes? The sex cells. So in males, they would be what? Sperm, and females? The eggs, the oocytes. Okay, so we talked before about mitosis, which is a type of cell division where we have one cell that divides to make two genetically identical cells. Okay, and this happens in all cells of the body when you're replacing skin cells and so forth. And now, how many chromosomes does a human cell have? 46. Okay, so that each one of those daughter cells would also have 46 chromosomes. Now, on the other hand, meiosis is a special type of cell division, happens only in the gonads, and here we produce cells that are haploid, have half the number of genes, and we also produce cells that are genetically different than one another. So we'll go from a cell that has 46, it will split twice to make up to 4 cells, each which will have how many chromosomes? What's half of 46? 23, okay. So they all have 23, and that's meiosis. The big thing is they're genetically distinct. Now, spermatogenesis is a type of meiosis that's going on in the gonads. We start out with a diploid spermatogonium. Okay, it undergoes mitosis, becomes a primary spermatocyte, then it keeps dividing under meiosis to eventually become spermatids, and then 4 spermatozoa. So we get a total of 4 spermatozoa for each spermatogonium. It's pretty much a bargain. Big thing is that each spermatozoa is different than the other ones. Okay, now here are the number of chromosomes for some common domestic species. You do not need to memorize this. But what you do need to realize is if I ask you on an exam, you know, a goat has 60 chromosomes in a diploid cell. How many would a sperm cell have? 30, right? Just take it divided by 2. The other thing I want you to realize is, like, which one of these animals would you say is like highest on the evolutionary chain here?
Well, then including humans.
Yeah.
Yeah, okay.
Okay, humans. You're going to put humans there. Fair enough. Okay. It's not like humans have more chromosomes than other species. Like, look at the cattle. The cattle, if you went by that thing, you would think, well, cattle have a lot more chromosomes. They should, you know, be much more advanced. But it doesn't follow that. In fact, we see a reduction in DNA if we go up the sort of evolutionary tree. But it has nothing to do with the number of chromosomes. Okay, so let's go to oogenesis, which I didn't realize is actually a trendy boutique on the North Shore. If you go up past Haleiwa, there's a little place called oogenesis. Never been in there. So here we have an oogonium. It undergoes mitosis to make a primary oocyte, which is diploid, and then it goes through 2 divisions of meiosis. And the big difference here between this and spermatogenesis is we still get 4 cells, but one of those cells is a lot bigger than the other one. We get 1 functional ovum out of this process and 3 polar bodies. Now we call them polar bodies because when that cell divides, here's the cell before mitosis, or meiosis. It undergoes the first run of meiosis, and then boop, there's the 2 cells. We call it a polar body because that polar body is just a small cell on the pole of the other cell. And the whole purpose of this is to reduce the number of chromosomes. Okay? But the reason that we have unequal division of cells is that how big is a sperm compared to an egg? It's really small. It's like this. Boop. And so the point is that there's not much cytoplasm in there to sustain the developing embryo until it forms a placental connection. So we want to make sure that our egg or oocyte is as big as possible to have a little bit of energy and supplies in order so that once that egg becomes fertilized, it can have the energy to divide for a while before a placenta is formed. And so we're only forming these polar bodies to get rid of the extra chromosomes. Now, what would happen if we didn't undergo meiosis? Let's say that you're a human with 46 chromosomes, your husband has 46 chromosomes, you get together, you have sex. How many chromosomes is your baby going to have if you don't undergo meiosis? 92, right? Seems like a good thing, right? This is like, oh, he's got twice the amount of DNA. It's not a good thing. Can you think of any genetic disorders in humans that are caused by having too much DNA? Yeah, Down syndrome, okay? Too many chromosomes in that case with trisomy 21. So we want to make sure we reduce the number of chromosomes to half before we make our gametes. All right, we're going to talk about the male reproductive system first. Why? It's simple, right? Compared to the female reproductive system, what it has to do is relatively simple. And that's just a couple things. We have to produce the male sex hormone, which is testosterone. We have to produce our gametes, which are spermatozoa. We also have to produce seminal fluid. Okay? As it turns out, the majority of the volume of the ejaculate is not sperm. It's other stuff, and we'll talk about that other stuff. And we also have to accomplish intromission, which sounds like something in a movie theater, but here we're talking about getting the penis into the vagina. Because as it turns out, having a penis makes reproduction a lot more efficient. All right, let's look at the testes. They produce sperm and hormones. They're located outside the abdominal cavity within the scrotum. Why is that?
To keep it cooler.
To keep it cooler. For some weird reason, sperm can't develop at body temperature. They have to be cooler than that. So they're housed in the scrotum, which allows them to thermoregulate. They have the spermatic cord consisting of blood vessels, nerves, and also the vas deferens. Okay. And regulation of the testis, it's regulated by the anterior pituitary, right? Start out with a message. GnRH comes down from our hypothalamus, gonadotropin releasing hormone, goes to our anterior pituitary, which then will secrete what? FSH and LH. Now, LH is the important one here for stimulating the production of testosterone. So LH travels to the testes. The Leydig cells of the testes will then produce testosterone. Okay? And testosterone will have a negative or positive feedback on GnRH and LH. Negative feedback. Most hormones are regulated by negative feedback. FSH is responsible for the stimulation of sperm production, and that happens within the seminiferous tubules. And there's also an inhibitory hormone called inhibin that feeds back on the anterior pituitary when we have adequate numbers of sperm. Okay? But all I really want you to know is be able to trace this hypophyseal gonadal axis from GnRH all the way down to testosterone. Okay, let's look at the spermatic cords. We already said that they contain blood vessels, and they also contain nerves and are vas deferens. And so one of the unique features of the circulation in the testis is what we have, this like countercurrent exchanger, which is called the papiniform plexus. So blood coming down into the testis is warm because the body temperature is warm. But blood that's returning to the body from the testis is cool because it's cooled off. So what happens is the two blood vessels run together, the artery carrying warm blood down and the vein carrying warm blood up, and they exchange heat on the way so that the warm blood goes right back up to the body, and the blood that arrives at the testis is a lot cooler. So this helps to keep the testes cool because, again, they need to be cool in order to produce sperm. Now, back in the 1980s, which is before most of you were born, there was actually some issues going on with people wearing really tight pants. As it turns out, in some places in Europe and stuff like that, it was actually leading to a reduction in sperm count in some men. And so they had to have these things out where urologists were telling men, stop it with the tight jeans, stop it with the tighty-whities, because that can actually reduce the sperm count. All right, we'll talk more about the screw-up in a minute. So first of all, the testes contain seminiferous tubule, which is a site of spermatogenesis. There are literally meters and meters and meters and meters of tubules within each testis. They're very long. And in there we have different cells. We have interstitial cells, which produce testosterone, Sertoli cells, which help to support the developing spermatozoa and spermatids. And so here's a section through the testis. And here you can see these seminiferous tubules. Now remember, the tubules go back and forth, so this tubule could be connected to that tubule. And you can see that the inside of the tubule we have a hollow lumen, and those tails that you see are the tails of the maturing spermatozoa. The meiotic cell division starts out here, so the most immature cells are located towards the outside of the tubule, and the most mature ones are located towards the inside. And if we look in between the tubules, we have these things called interstitial cells. Remember, interstitial just means in between, so these little triangles are interstitial cells, otherwise known as Leydig cells. And those are the guys that secrete testosterone in response to LH. Okay, here's just a close-up of the wall of that seminiferous tubule. You can see that the most primitive, the earliest cells are located towards the outside of spermatogonia. They undergo mitosis to become a primary spermatocyte. And then the other divisions are meiotic, and we produce our spermatids and eventually spermatozoa. Spermatids have a lot smaller heads than anything else, and the spermatozoa have very small heads as well. So at the end of this process, we're going to undergo spermiation, where that sperm is actually released into the tubule. Okay, we already talked about our non-spermatogenic cells, or Leydig cells, which are located outside the tubes that secrete testosterone. These are the Sertoli cells, also called the mother cells. They nurture the developing sperm. And the other thing they help to do is create a blood-testis barrier that shields the testes and the sperm from the immune system. And that's important because remember, the cells, the sperm cells, actually have different markers on them than a normal body cell. I mean, genetically, if your immune system could get a hold of these cells, they'd attack them because they don't identify them as your own body cells. And so somehow, the cells in the testis, along with the Sertoli cells, form this blood-testis barrier that prevents inappropriate immune response. Okay, if you look at the sperm themselves, they're not much more than just a capsule of male DNA. So we have our genetic material, and here are our chromosomes. The other thing that's in the head is we have a little bit of hydrolytic enzyme. Because when it comes to the egg, you would think if you're the first sperm to get to an egg, you would be like the winner. You're like, woo, this is me, I'm going to fertilize the egg. Not true. because every time a sperm comes down, they're going to barf up a little hydrolytic enzyme, and it's going to eat away through the covering of that egg just a little bit. As it turns out, it takes probably 50, 60, sometimes several hundred sperm dropping off their hydrolytic enzymes to burrow enough, deep enough in that egg so that the next sperm can drop off its genetic information and fertilize that egg. Okay? So that's why we have this acrosome with the hydrolytic enzymes. The mid piece here has lots of mitochondria, which is important because sperm swim a lot, so they need a lot of sources of energy. So this helps them extract energy. And then the tail also contains our contractile motor fibers that help them to swim. There's not really much energy stored in the sperm per se, so they're getting their energy from where? The semen, the seminal fluid. And the majority of seminal fluid are things like glucose. There's some prostaglandins in there. There's a little bit of alkaline material in there. So the glucose in there is helping to nurture the sperm and help them, give them an energy source so they can swim. Okay, going back to the scrotum, that was that loose sac of skin surrounding the testes. It helps to thermoregulate the testes, keep them a little bit cooler than body temperature. And this is done by something called the cremaster muscle, okay, which is connected to the skin. And if it's really hot outside, it will sort of dilate and allow the testes to hang down. If it's really cold, it will constrict and bring up the testes closer to the body. Okay, now the interesting thing about the testes is they actually originate in the same place as the ovaries within the body. They start out up by the kidneys. And so as that animal grows and develops, a muscle called the gubernaculum will help bring them down, okay, through the inguinal canal or inguinal ring and place them in the scrotum. And it's really important because, right, we can't produce sperm if that testis remains inside the abdominal cavity. And in some species, that could actually become cancerous if we can't bring that testis down. So it's important that we have the gubernaculum to bring those down. Now, interestingly, some animals like rabbits can actually retract their testes during a fight, which seems like a really cool thing, right? But unfortunately humans haven't had that happen. Okay, the outside of the testis, main thing I want you to know is this structure right here is the epididymis. This is where sperm are stored prior to ejaculation. The sperm in the testes aren't ready to be ejaculated yet. Even though they might be full-blown sperm, they actually have to mature and be stored here in the epididymis. Now, occasionally, a testis doesn't descend fully during the animal's development. So we get something called cryptorchidism. Okay? It's basically hidden testis. And so the testis, again, starts out by the kidney, and it should descend through the inguinal canal to end up in the scrotum, but sometimes it gets stuck. And so if it gets stuck, you might have to go up there and surgically remove it, which is usually a good idea. It can continue to produce testosterone, but it can't produce any sperm. But again, most veterinarians, if they detect only one testis in there, will recommend going in, getting that second testis, and surgically removing it. Okay, the vas deferens is the tube that helps to carry the mature sperm to the urethra. It also helps to carry the seminal fluid. So there's a little ampular duct that empties into the urethra after all the seminal fluids are added. Okay, so we have lots of accessory reproductive glands. They're not necessarily all in each species. The three glands I want you to know about are the Cowper's gland, otherwise known as the bulbal urethra gland, the prostate gland, and the seminal vesicles. Again, not all species have all three of these. These are actually making the majority of the seminal fluid. So in addition to glucose, we said the seminal fluid has prostaglandins, and it also has alkaline material. It has a buffer in there. Why do you think we need a buffer in our seminal fluid? Yeah, but why does it need to be basic?
Oh, because the female's body is acidic, that's how.
Yeah, the vagina is a very hostile environment. It's very acidic. And that acidity is good because it helps to protect the vagina and the reproductive tract, but it could be potentially fatal to the sperm. The other thing is, think about what tube these sperm are going to travel through before they get to the vagina. What's the name of that tube? The urethra. And what else has been going through that tube? Urine, which is acidic. So the bulbourethral gland, or Cowper's gland, actually has what we call pre-ejaculate that is also alkaline and will neutralize any leftover urine in the urethra prior to ejaculation. So that's really important. And the last thing that's in there are prostaglandins. Okay, what do prostaglandins do? No, they're actually hormone-like compounds that can stimulate smooth muscle contraction. So the prostaglandins in there can actually stimulate uptake of the seminal fluid by the female reproductive tract and help to transport it to the uterine tubes where fertilization occurs. In fact, you don't think about this. You think about it happening on a microscopic level, but I remember we did some artificial inseminations with some sows out in Waimanalo, and we had about 50 mils of seminal fluid that was diluted for each sow, and you put it on this little bitty, I guess, catheter that has like a corkscrew at the end, because a pig's penis is sort of corkscrew, and you put it in the pig's vagina, and then the guy would come and he would pet the pig and sort of, I don't know, just get it in the mood, and all of a sudden you would see, you didn't really have to squeeze this container, it would get sucked into there. Okay, by this peristalsis. It was freaky to see. So hopefully maybe your class will be able to go back out and see that. But there is a little bit of reverse peristalsis in some animals in the reproductive tract that helps to draw the seminal fluid in. Okay. Now the urethra, again, is just a tube through which urine is flowing. It's also a tube through which semen is flowing. There's a pelvic portion, which is inside the body cavity, and a penile portion. And so, yeah, it's important that we have an open, patent urethra in order for ejaculation to occur. All right, then we get to the penis. I said before that penis is a good thing. Not all animals have a penis, but it makes reproduction a lot more efficient. So can you think of an animal that does not have a penis?
Like lion or like a sea animal?
Like what?
Like a sea animal? Or
Sure, anything.
like an anemone?
Yeah, an anemone doesn't have a penis. Okay.
Or fish?
Yeah, most fish don't have penis. So if you look at a fish and the size of its gonads, they're relatively large in proportion to their body size. This is actually fairly small. But if you looked at a gravid reef fish like we have out here in Hawaii, they're about this big. So in a human male, it would be like having each testis weigh 7 or 8 pounds. Okay, and the reason is because a lot of the sperm go to waste. Okay, when you talk about a female fish swims by, she may, shed her eggs, and the male will go up and just like, wah, and and ejaculate in the water column. But the majority of sperm never reach an egg, and the fact that the survivorship of those gametes is so low anyway means they just have to produce a lot more of them. So by having a penis, mammals tend to have a lot smaller testes because penis makes things more efficient. Now, that being said, dogs have really weird penises. They're freaky. So we have a shaft on there and the root and the crus that help to secure those into the abdominal cavity. And then the glands out here with lots of nerve endings in there. And it's all surrounded by a prepuce, which would be similar to a foreskin in a human. Now, the interesting thing about dogs is that they have this bulb of the glands, or bulbus glandus, which is actually in back of the long part of the glands. And this is the part that becomes erect after prolonged stimulation. Usually it doesn't become fully erect until after the animal has entered the female and ejaculated. And this is what holds them there, right? Have you ever seen two dogs get stuck together? Yeah, and it's funny because they're just like, and they turn around and they can't go anywhere for a while. So we think it's important that holding those two animals together allow for more time for the semen to go in the vagina. And of course the penis itself is filled with erectile tissue. We have a corpora cavernosa and also a corpora spongiosum. Okay, the spongiosum surrounds the urethra, holds that open, whereas the other two just help to make the penis erect so it's easier to enter the female. All right, now there is some weird variation in penis shapes among animals. If you look in our former lab book, they had pictures of, again, what it would look like in a bull, what it would look like in a cat. Cats in particular are kind of weird. They've actually got these recurved spikes on them, or hooks. So you can understand why when you hear cats out yowling on the fence post or something like that, that it may not be that fun to have something with hooks inserted. In any case, we talk about things such as the ram, the bull, and the boar. They actually have a sigmoid flexure in there. So in most animals, the penis just gets, more erect and maybe a little bit longer. In the boar, there's this weird thing to where it's retracted on a ligament, but when blood is injected in there during stimulation and arousal, it will actually cause this whole thing to straighten out. Your book compares it to like a fire hose that's all kinked up, and then you pressurize that hose. What happens to it? It straightens out. So that's exactly what's happening here. And then once the erection goes away, there's another piece of tissue in there, muscle tissue and connective tissue that help to draw it back inside the body. Okay, erection and ejaculation. Erection is a parasympathetic process, okay? What things do animals find arousing? Smell, okay? A lot of it's based on olfactory cues. Some of it could be visual or even auditory. For the most part, it's smell. Has anyone in here done an artificial insemination? On a dog? Yes. Bulldog?
No, dogs.
Oh, what kind?
Livestock. Cows.
Cows, okay.
Pigs.
Oh, pigs.
Sheep.
Sheep? Oh, okay. I had to do it on a bulldog. And so you held the female bulldog in front of the male so he could smell what's going on there and to get him all randy. And then the vet was sitting back there on the male and, doing his thing. And eventually they get it to ejaculate into a tube. And then he went and counted the sperm on the slide, make sure that it was enough sperm and that they were motile. And then we injected him with a catheter or something into the female's vulva. And then I had to hold the female in this reverse U to make sure that it would go down to the right place to the uterine tubes and stuff like that. So yeah, that's one of those things that you might get to do at some point. It's kind of, they tend to do it to the new guy, so just be warned. All right, so erection is a parasympathetic reflex, okay, involving different cues for arousal. Now, when erection happens, basically it's because the arteries are bringing blood faster in than the veins are draining it. The veins actually get pinched off in the process of the erection, so the blood just engorges the penis, it has nowhere to go. Ejaculation is a sympathetic reflex. Think about, what do you normally think about with sympathetic reflexes? Fight or flight. So there's like, it's just like, traumatic or something like that. But that's going to cause semen to be ejaculated into the urethra, out of the penis and the female reproductive tract. Now, once it's there, remember that sperm can be active for about 48 hours. And that's pretty weird to think about that, they're vital for that long. Okay, the other thing is just remember that there are some prostaglandins in there that may actually stimulate contraction of the female reproductive tract as well. Okay, so at the end of this, you might see something like this, which is called a tie, right? The dogs are stuck together, and what structure is holding them together? Bulbus glandis. Like, you might even notice too, if you ever have a neutered dog and they're like having a good dream or something like that, and all of a sudden this thing appears, my wife's like, he still has testicles. I'm like, no, he doesn't, but it's the bulbus glandis will still enlarge sometimes. I don't know, when they're thinking about stuff, I guess.